Hello, students, and welcome to my 2023 video on photosynthesis and cellular respiration. This is designed for the students here at TASM, but anybody that stumbles across this on the web can use it. Just like all my videos, it is not for profit. It is only meant to help students, and this will cover the carbon cycle, photosynthesis, and cellular respiration. Here's a picture of my cat climbing up a tree that's doing photosynthesis, and the cat's doing a lot of cellular respiration. Haha. Let's jump in. All right. So we did our first unit was about sea turtles and carrying capacity and ecosystems. And now we've moved on to photosynthesis and cellular respiration. And if you remember back towards the beginning of it, it's been a pretty long unit, but we tried to tie it together with coral bleaching, right? And so we said that, sticking in an aquatic ecosystem, what's happening to the corals around here, and we're starting to see this happen in the Dominion Islands here and Bandar Kairan in Muscat, Oman. So how do we connect Unit 1 and 2? Unit 1 was about ecosystems, like I just said. Unit 2, the history of Earth's atmosphere, photosynthesis and respiration. And we're connecting them via the carbon cycle. We take in sugars and oxygen, which are produced by autotrophs or plants, and we give off carbon dioxide and water, which plants use to grow and make their own sugar molecules. How should you use this video? You should pause the video often, see if you can answer the questions. You should try and construct models of the material. I've given you the origami organelles. We have light-dependent reaction models here in the classroom. You should try and construct explanations of everything and then explain them to your parents. For example, look at this. You shouldn't just try and memorize this. You should say, oh, okay, here's the model. The model's showing me photosynthesis because I see water coming in, oxygen going out, CO2 coming in, and making sugars. And then I can see that within photosynthesis, there are cycles, the light reactions and the Calvin cycle, and how one makes products that the other one uses as reactants. All right. Let's dive in. You did some really cool gizmos, by the way, on coral bleaching and then on cyanide and how they affect the systems. And I will ask you questions in your assessment given this model, and then with, say, the addition of diuron or paraquat as a herbicide, or the addition of cyanide to a model, how has it changed? What are the effects? Use the resources that work for you. Use the Google Classroom. You've got everything's underneath Unit 2. And then I've also posted, various, this video and the study guide and practice problems underneath the assessment tab. Okay, let's go ahead and start. Why is photosynthesis and cellular respiration called a cycle? Pause the video and answer. Okay, hopefully you got something along the lines of the products of one are the reactants of the other, or you could say the outputs of one are the inputs of the other. Either of those are acceptable answers. Here we can see this nice little graphic from the Amoeba sisters. Let's look at what goes into photosynthesis, right? We'll start with photosynthesis. It means to make from light. And so here with carbon dioxide and water and light, the plant's going to be able to make sugars and oxygen. How lucky for us. We then take those sugars and that oxygen. This would be heterotrophs and autotrophs as well. So us, trees, dogs, everything has to do some type of cellular respiration to get energy. So we'll take that sugar and that oxygen, and then through a series of reactions, we'll make CO2, which is carbon dioxide, water, and energy. You can think of ATP energy as the form of energy we use that all life on Earth uses. Okay, here you can see the cyclical nature of it. So the plants are taking in carbon dioxide and water going this way into the autotrophs. That can also include bacteria that are photosynthetic. They're going to produce sugars. That's what organic compound stands for, and oxygen. We then consume that as animals or fungi and other bacteria as well, consume those sugars and oxygen and produce carbon dioxide and water. With those sugars and oxygen, we make lots of ATP, or energy. Here's a slide that I had from my first year teaching. So this is all the way from 2004. Shout out to those students. I hope they're doing very well from East St. John High School. Here, the sun shines down on plants or autotrophs. They engage in the process of photosynthesis to make glucose. That makes glucose and oxygen. Plants and animals, right, then engage in cellular respiration to break down that glucose into carbon dioxide, water, and energy. This is a tricky one for students to remember, that plants also have to do cellular respiration. The model I said in class is that glucose would be like a 50 real note. And for those of you not in Oman, it'd be like $100 bill. It's a big denomination. And then ATP is like a single real note or a single dollar bill, and it's easier to use. It's easier to go around and buy stuff with it. All right. What are the word equations and chemical equations of photosynthesis and cellular respiration? I'm not going to ask you to spit out the chemical equation on the test, but it might be embedded in a multiple choice question for you to figure out. Here we go. Hopefully you got something along the lines of this. I do expect you to know the word equation: carbon dioxide and water and light yielding glucose and oxygen for photosynthesis. For cellular respiration, glucose and oxygen yielding carbon dioxide, water, and energy. All right, it's a cycle. Now, can you pause the video, rewind, and show how that equation is balanced, right? How is this balanced? Go ahead and pause the video. Here we go. Let's check it out. We're going to count the atoms. I only showed this to you once in class, and I did not do a lot of guided practice with it. So let's try it here. If I'm looking at this one up here, I'm seeing 1 carbon. Here I'm seeing H2O. I'm seeing 2 hydrogens. And the trick here is I need to add the oxygens in both molecules. I'm seeing 2 oxygens here. And if there's no number, it's understood to be 1. So I'm seeing 1 here. 2 plus 1 is 3. On the other side, I'm seeing 6 carbons, 12 hydrogens. And then how many oxygens? I'm seeing 6 here and 2 here. That would be 8. So this is not balanced, right? And that violates laws of nature. So let's see what it is balanced. Balanced here, if we had a 6 in front of the C and a 6 in front of this H, it's going to come out balanced. Now I multiply the coefficient times the subscript, so 6 times 1, 6 carbons, 6 times 2 for 12 hydrogens, and the oxygen is the tricky one. Once again, I need to multiply for both molecules and then add them together. So I need to use my order of operations. 6 times 2 is 12. 6 times, if there's no number there, it's 1. 6 times 1 is 6. 12 plus 6, I have 18 oxygens amongst these 2 molecules. You could draw it out. Like seeing my children do math in elementary school, they often draw out arrays and other visual representations. So you could draw it as well. And so if you actually drew 6 CO2 molecules and 6 water molecules, you could count them up and you would see you'd get 6 carbons, 12 hydrogens, and 18 oxygens. If we look at this side, it comes out, it's a little easier to do here. If we look around, we see here's the carbons, we got 6 of them. Here's the hydrogens, we got 12 of them. Here we have 1 as a coefficient. If there's no number there, it's understood to be 1. So we're going to have 6 oxygens right here. And then here we have a 6 and a 2. So 6 times 2 is 12. Add them together and we get 18. Now this equation is balanced and it shows, in essence here, photosynthesis. All right. How does ATP provide energy? So if you pause the video there, that's great. I'm sorry, I forgot to tell you to pause. This green molecule is called an adenine. You don't need to know that yet. We'll learn that later on. But it's a nitrogenous base. It has nitrogen in it. And it's connected to a sugar. This is the sugar. It's called ribose. And then it's going to be connected to either 1, 2, or 3 phosphate groups. And if you have 3 phosphate groups, I want you to think about that as your cell phone fully charged, 100% battery, good to go. If you remove one of those phosphates, it becomes ADP, and it has now become half charged. You're at like 50%. You've used the 50% in your phone to do stuff, to do Snapchat, to send messages, to do Instagram, right? So when we lose a phosphate from ATP to ADP, that energy is transferred to somewhere else. For instance, it might be helping me to blink my eyes right now. We can recharge ADP, just like your phone, it's reusable. We can add a phosphate back to it in the process of ATP synthesis. Occasionally, we might get all the way down to one phosphate, and that would be called adenosine monophosphate. Here's the cycle. We take the energy that we get from food that we ate, we break that down in a process called catabolism to make ATP. We then use that ATP to do work. Well, what kind of work might we do? Some work that we might do might be including cells dividing, that takes energy, transcribing genes, that takes energy, running around takes energy, breathing takes energy, running different pumps in our body, like the sodium potassium pump, takes energy. So we use that to accomplish the items we need to be alive. Once we use up the energy, it becomes ADP in a phosphate that's floating around. And then we use energy from food to remake that into ATP. Here's another good example of it. ATP is a charged battery. You take off a phosphate, it's going to release energy. The battery will be half charged now. We then take energy from food to recharge it. We can use it again and again and again. All right. Why is photosynthesis so vital to us? Where does the mass of a tree come from? All good questions that hopefully you should be able to answer now. So why is it so vital to us? Well, it's able to take solar energy and convert it into sugars that we can then use for life on Earth. If photosynthesis wasn't around, we wouldn't be around. We wouldn't be able to survive. There are some organisms that can use energy at the bottom of the ocean at hydrothermal vents. But the vast majority of life relies upon photosynthesis and solar sunlight energy. All right, I believe in you. Aha! What's in a leaf? How do plants get the reactants they need for photosynthesis and release the products? Well, plants have roots and stomata. In the roots, that is where water comes in, and then certain chemicals that the plant will need for photosynthesis. So water is the big one that we'll worry about in our introductory biology class. It gets in at the roots. And then in the stomata, which are the little holes or openings in the leaf, I'm going to go get my model right now. Okay, I'm back. Here's our model, which is older than you all. Here's the stomata, or the opening that oxygen and CO2 are exchanged through. Right, and then in class we actually sucked all the air out of here and then tried to watch it do photosynthesis. These cells right here are called palisade mesophyll cells, and they're going to be the area or the cells that do the most photosynthesis. So let's zoom in on the actual organelle that does photosynthesis. An organelle is a specific function, or a specific item in a cell that performs a function. For example, like an organ would be like your heart to pump your blood, or your lungs to exchange oxygen and carbon dioxide. Here, the organ or the organelle would be the chloroplast to engage in photosynthesis. Here is the chloroplast, and so there'll be a lot of these in a plant cell. The small green pancakes are called thylakoids, and that's where the light-dependent reactions of photosynthesis will take place. A stack of thylakoids is called a granum, and then multiple granum together are called grana. The liquid inside of here is called the stroma, and that's where the Calvin cycle will take place. All right, why are plants green? So we got to do this in a couple classes. It didn't work super well, but we took some leaves, we grounded them up, or in some classes I put the chlorophyll extract on the paper, and then you added alcohol to them. And you were able to see that the plants had pigments inside of them. The most efficient one is chlorophyll A, but plants also have other light absorbing pigments in them, such as carotenoids and xanthophylls and chlorophyll B. That helps the plant capture different electromagnetic wavelengths or the light. But if I look at this graph right here, I can see that we absorb blue light, absorb red light, and orange light, but not really green light. And that's why plants are green. It's not absorbing that color and it's reflecting that color to our eyes. So, since a plant is not absorbing green light, if we were to grow a plant underneath green lights, it would not do very well. It would die. All right, and we tried that in one or two classes. This lab unfortunately just didn't work very well. But I went to a conference, as you know, and I'm gonna try and get algae beads next time. I think those will be pretty cool. All right, can you model the cycle of the light-dependent reactions in the Calvin cycle? On your test, I will give you a word bank, and then we will mutate the model as well. But you will have a word bank. So I'm looking at it. Let's go ahead. Number one, this is the stroma, the liquid in here. Number two, that's a thylakoid. So we're looking at the structures. And so the light-dependent reactions, okay, that depends upon light. And in the light-dependent reactions, what happens is at the thylakoid membrane, we're going to take in water. We're going to produce oxygen. Take in water, produce oxygen. Therefore, all the oxygen we breathe comes from water that has been split by plants. We're on to number 5. That's going to also produce ATP and NADPH. ATP, from previous part of the video, we know is energy that the plant can use. NADPH is a new term for us, and I have said this in class a couple times. You can think about it as an electron carrier. It's carrying 2 electrons and 1 H plus. You could think about it like a taxi cab. It's carrying 2 electrons in the back seat, 1 H plus in the front seat, and they're going to the Calvin cycle. The Calvin cycle is going to take in carbon dioxide from the atmosphere. It's going to use the energy from ATP. It's going to use the energy from the electrons and the H plus here to make sugars. So once again, the Calvin cycle is using, through the process of enzymes, it's using the energy from ATP and from NADPH to make sugars. It's then going to send back those used up items to the light-dependent reactions. Here is that half-dead battery, the ADP and a phosphate. And here is an empty taxi cab. It has no high-energy electrons and no H+. That's NADP+. I would encourage you to model the light-dependent reactions. Unfortunately, I wasn't able to print this in color for everybody, but I do have extra copies of this if you'd like to play with it. So let's go over. Remember that the word bank will be provided on the assessment for the light-dependent reactions. I found this interesting, and I think your Gizmo did an excellent job of going over this and then showing you how changes to the system affect how the plant works. And so I would encourage you to look back at the Gizmo and also to try this right now by pausing the video and going through it. All right. This is the stroma on the outside. So that's the outside liquid. This is the thylakoid membrane. Here at the membrane, let me use my pen. This is called a phospholipid bilayer, and the inside of it is very nonpolar, and it repels anything that has a charge. So an H plus might go up to it, but can't get through it. No dice. It has a charge. Can't get through it. But luckily for us, what can get through it is oxygen. O2 really looks like this, an oxygen bonded to another oxygen, and it doesn't have a charge. And so it's able to just float on by, and it goes from an area of high concentration to low. And so that was the stroma. The inside is called the thylakoid space, is what I used in your papers, also called the thylakoid lumen. So this is inside the thylakoid. What happens is light strikes photosystem 2. Notice how these are out of order. It's just they actually discovered this one first. Photosystem 2 is a protein complex that contains chlorophyll. The chlorophyll will get excited by the light and it will lose an electron. That electron will go on this pathway called an electron transport chain. It can't just go anywhere. The electron can't go out here. No. It follows this chain. The electron from chlorophyll must immediately be replaced. And the way it's replaced is through an enzyme that promotes the photolysis or the splitting of water. From the H, the hydrogen in the water, the hydrogen has one electron and one proton. The electron is ripped off and taken into photosystem 2. The proton is now left here, and that is an H+, and it is in the thylakoid space. Another way that we get more H pluses in the thylakoid space is that as the electron passes through this chain, and as it passes through this transmembrane protein, this protein that goes through the whole thing, it's able to pump H pluses into the cell. This is against their gradient. Notice how there's only 2 H pluses out here, or I guess you could say 3. And there's 1, 2, 3, 4, 5, 6, 7 in here. They're going against their gradient. It'd be like putting perfume back into a bottle. So the electrons have now lost their energy. They're at photosystem 1. What did they do here? They did 2 things. They pumped H pluses into the thylakoid space, and to be replaced, water was split in a process called photolysis, which gave us H pluses and oxygen. The electrons at photosystem 1 are excited again. They get another bolt of energy from light, and they go on another electron transport chain to an enzyme that's going to add them to NADP+. So let me draw out NADP+. NADP+ looks like a taxi cab that has 3 open seats. NADP+ is the empty cab. NADPH is when the seats are filled. So now we're going to add an electron. Oh, that's a janky electron. An electron and an H+. And students, now we have, excuse me, NADPH. This NADPH goes off to the Calvin cycle to help make carbohydrates, or to help make sugars. Now we have lots of H pluses inside the thylakoid, lots of H pluses in here. They can diffuse through ATP synthase. ATP synthase is this enzyme right here. It's like a one-way street. This one was also one-way. For every, I believe it's every 4 H pluses that go through here, it will turn the rotor and it will add a phosphate to ADP to make ATP. So once again, we then send this ATP to the Calvin cycle. So you should go back and try and explain how the light-dependent reactions make NADPH and ATP, which are 2 inputs for the Calvin cycle. The Calvin cycle gave the light-dependent reactions NADP plus and the ADP and the phosphate. So notice how the products of one are the reactants of the other. If you're a little overwhelmed right now, this is definitely challenging, but it's a good challenging. Now, you might say, what in the hexagon is the point of learning all this? And you're right. I mean, it's challenging stuff, and it's going to prepare you for AP Biology, which will be great, and I hope you take. But at the same time, we could also look at this and see what happens if part of the system is mutated. Let's say that photosystem 1 gets shut down. Well, then what will happen to the system? How does that kill the plant? All right, let's clear the screen and move on. Here's another picture of it. So you could pause the video here and see if you can go through the process of the light-dependent reactions. Here's the big picture again. Notice how the light-dependent reactions in the thylakoids are making NADPH and ATP, which will go to the Calvin cycle, which will then make sugars and send us back NADP plus and ADP. Okay. The Calvin cycle is definitely challenging. For some of you all, I showed you a TED Ed on it, and that was definitely challenging. I'm going to go over it, but it's not going to be featured heavily on the assessment. Big picture, what you need to know is that the Calvin cycle makes sugars. And it's going to take in carbon dioxide. It's going to take the energy from ATP, the H's and the electrons from NADPH, and it's going to make sugars. It occurs in 3 steps. There's fixation, reduction, and regeneration. In fixation, there's an enzyme called Rubisco, and you can see it centered here. Rubisco is going to be able to pick up carbon dioxide and attach it to a pre-existing molecule that's called RUBP right here. So it hooks the carbon dioxide onto RUBP. That's going to split into 2 3-carbon molecules that can then be worked upon to make the sugars. So the important part of carbon fixation is getting CO2 to be fixed or added to this molecule. It's making CO2 become part of a sugar. In reduction, the important part here is that NADPH loses electrons and hydrogen protons, and they're added to this molecule that's made from fixation. That's going to become glucose. The energy here is rearranging bonds as it's making the sugar. In regeneration, the starting molecule is made again so that the cycle can continue indefinitely. All right, this is more efficient than just combining together a whole bunch of CO2s. So it's nature's way of making sugars. All right, some factors that affect photosynthesis. Carbon dioxide. We're putting more and more carbon dioxide into the atmosphere. Is that helping or hurting plants or not leading to a change? Temperature. The temperature on Earth is rising. Is it helping or hurting plants or not leading to a change? Light intensity. How intense the light is on it, or how many photons are hitting it per second. And then the wavelength of light or the color. All right, we did an online lab to check this out, so you could try that again as well. Can you construct a graph for this? I won't ask you to construct a graph from scratch. I would either give you data or I would give you graphs and ask you to describe what's going on. So let's look at these. Can you describe what's happening in each graph? If we look here, for light intensity, we see that to a certain point, increasing light intensity increases the rate of photosynthesis. Maybe that's 100 watts. Once you get past that, it kind of levels off. And so what is going on there? It means that the light-dependent reactions can only go so fast, right? They would be the limiting factor here, the limiting rate factor. Photosynthesis can't speed up any more than that. For this one, can you describe what's going on? So you add more CO2 to the system, it's going to increase photosynthesis till you get to a certain point. Once you get to that point, adding any more CO2 won't speed up the process of photosynthesis. Rubisco simply can't go any faster. So giving it more reactants won't make it go any faster. It's kind of like your relay team for track, right? You gotta you can't make it any faster. There's going to be I guess maybe that's not a great example. Let's use the example of a factory making phones, right? Everybody loves phones, etc. So it can only go as fast as the slowest step. You can't speed it up past that. Temperature is a little different though. Right? If you get really cold, photosynthesis will slow down. The molecules will move less quickly. If we get really hot, it can actually denature the proteins in the photosystems. And so in your gizmos, you saw in the Great Barrier Reef, that it's getting so hot that sometimes it denatures the ability of photosystem 2 and 1 to work well, right? It's messing up those enzymes, those proteins. All right. Here's some plants. So if you take AP Bio, we'll get to play a lot with these and do transpiration labs. And we're getting ready to move on to cellular respiration. So go ahead and take a break. I'm going to take a quick one, and then we'll come back and crush this. All right. I hope you had a good break. Let's crush some cellular respiration for the mitochondria here. And, thinking about our powerhouse of the cell, like our gym right here at Taysom. We see so many memes about it, people bring it up in graduation speeches, etc., that the mitochondria is the powerhouse of the cell, but you're going to learn how it really works, how it really makes energy. Here's a big picture view of it. We're going to take sugars or carbohydrates or fats that we eat, and they're going to enter the cellular respiration pathway. Different items may enter at different points. For example, sugars have to be broken down into glucose. Like a big sugar, like say sucrose, is broken down into glucose, and it can enter here. Fats may enter in at different parts. All right, so here is glucose outside of the cell, brought to you by your blood. There's a receptor there that brings glucose into our cells. Glucose is like that 50 real or 100 real note or $100 bill. It's too big to use. We got to break it down into smaller bits, and those smaller bits of energy are called ATP. And so we're going to do this through a series of reactions that are going to give us the ability to generate ATP. And so the first one is going to be glycolysis. It's going to break down glucose into pyruvate. If there is oxygen available, that pyruvate will be transported to the mitochondria, to the matrix, the very inside part here, and then we'll go through the Krebs cycle, or the citric acid cycle. After the citric acid cycle will be the electron transport chain, which will then create lots of ATP for us. That's one way it can happen. Other products include carbon dioxide and water, and that's going to go to plants, right? The carbon dioxide is able to diffuse out of our cell. No oxygen present, then we'll engage in fermentation. And that fermentation could either be lactic acid fermentation or alcoholic fermentation. And we'll go over that later on. All right, let's dive in. Can you model or describe glycolysis, the Krebs cycle, the electron transport chain? 1 second here. Okay. Glycolysis is going to happen on the outside of the mitochondria in the cytoplasm or the cytosol. In glycolysis, the word lysis means to break, and glyco stands for sugar, so we're going to break the sugar in half. And so here you're going to take glucose. You're going to use 10 enzyme-mediated steps to make 2 ATP, 2 NADH, and 2 pyruvates. Those pyruvates will then enter into the matrix of the cell right here, the matrix of the mitochondria, and undergo the Krebs cycle, named after the person who discovered it. The Krebs cycle takes all the useful energy off of pyruvate and puts it into NADH and FADH2, electron carriers. These will then go to the electron transport chain. The waste product, which is CO2, is made during the Krebs cycle, and CO2 can diffuse out of the mitochondria and eventually out of the cell. The electron transport chain will take those electrons, it will take oxygen that you breathe in, and it will then make water, lots of ATP, and it will empty the taxicabs. It'll empty the NADH and the FADH2. The real term is called oxidizing them back into NAD plus. In FAD. Let's see each bit in a little more detail. Here is glycolysis. Glycolysis takes a little bit of energy to get started, and then it creates a net gain of 2 ATP, and it split that glucose molecule into 2 pyruvates. Here's a good picture of it. So the inputs are going to be glucose and ATP. The outputs and NAD plus. So glucose, ATP, NAD plus, the outputs are going to be a net gain of 2 ATP because it made 4, and then it's going to give us 2 NADHs as well as 2 pyruvates. Once again, let me draw what I want you to think of NADH as. Just like in photosynthesis, I want you to think about it as a taxicab that's got 3 seats. So this would be NAD plus. That's when it's empty. Now let's add some electrons down and 1 H plus, and it becomes NADH. Notice how there was no oxygen required for this. And so glycolysis evolved very early on when there was no oxygen in the atmosphere. Here's the Krebs cycle. The Krebs cycle is pretty complicated. It's well, so is glycolysis, I guess. It's got 8 enzyme steps that you don't need to know, but I would like you to think about what are the reactants, what's coming into this and what's going out. Well, if I think about what's coming in, I see pyruvate was coming in and it was converted to acetyl coenzyme A. We went over that with your escape room. That comes in through a bunch of reactions. It's going to make lots of NADH and FADH2s. NADH we already went over. FADH2, you can think about it as having an extra seat in the cab. And so here, 4 taxi cab seats. And we're going to have electron, electron, H plus, and H plus. All right. So we've made a lot of electron carriers, right? And we didn't just make them out of nothing, right? They were there. They were there as NAD plus, as empty ones. So we filled them up would be a better way to describe it in the Krebs cycle. The byproduct of all of this is going to be carbon dioxide. And this carbon dioxide is going to diffuse out of the cell, and we're going to breathe it out. All right. On to the ETC, the electron transport chain. This is awesome. Here at the electron transport chain, once you understand photosynthesis, it's pretty similar. In photosynthesis, the source of the electrons was water. Here, the source of the electrons is going to be NADH and FADH2. So NADH is going to drop off an electron. FADH2 is going to drop off 2 electrons. And they're going to go on this electron transport chain. All right. These electron transport chains will go through protein complexes. This is number 1. This is number 2. This is number 3. And this is number 4. And as they go through these these protein complexes, they're able to pump H pluses into the intermembrane space. If you're wondering where the intermembrane space is, here's a mitochondria. This is the intermembrane space where the pink is. This spot in between the outer membrane and the inner membrane. Remember, just like in photosynthesis, the H pluses can't get through this membrane. It's impermeable to them. Anything that's charged cannot get through. The H pluses are able to diffuse through ATP synthase here. They go through rapidly. And so for every 4 that goes through, it makes an ATP. It turns this rotor and adds a phosphate to an ADP. Now, where did the H pluses come from here in the matrix? They came from NADH and FADH2 as they dropped off those electrons. Now, something has to get rid of all the electrons at the end of this chain here. And so to get rid of them, they are accepted by oxygen at an enzyme called cytochrome oxidase. They're going to be accepted by oxygen, and they're going to combine with H pluses here to make water. So H pluses, oxygen, and electrons all come together to make water, and this is why you have to breathe oxygen. You have to be able to clear out the electrons from this chain. If you don't breathe oxygen, this chain shuts down, and that's actually what kills you if you don't breathe oxygen. All right, in your gizmo, you saw that cyanide shuts this down. And in class, we talked about dinitrophenol, briefly, not on the test or anything. But there's an antibiotic called oligomycin that shuts down this. Dinitrophenol enables a proton uncoupler to come right here. It enables the movement of H plus through without having to go through ATP synthase, which means that you're not making enough ATP and you'll keep doing cellular respiration because you're desperately trying to make ATP, and so you'll burn up all your fats. But then it leads to people dying because it's too hot. This friction from the H pluses coming through here can raise your body temperature to the point that you die. Notice how once the NADH and FADH2 drop off their electrons, they now become NAD plus and FAD again. They're like an empty taxi cab. They're ready to go back to the airport and pick more electrons and hydrogen protons up. They're ready to go back to glycolysis and the Krebs cycle. So here's another good little overview of it, right? If we have glucose in our cell, it will then undergo glycolysis to become pyruvate. This is an anaerobic process, does not require any oxygen. If you have oxygen present, it'll go to the mitochondria and go through the Krebs cycle and then go through the electron transport chain. That's what oxidative phosphorylation stands for here. Oxidative means that this is losing electrons, and phosphorylation means that we add the phosphate to ADP. Let's say that there's no oxygen present, then there will be anaerobic respiration where it will produce either lactic acid or ethanol. So let's go ahead and jump into alcoholic fermentation real quick. Without oxygen, we do repeated rounds of glycolysis in an effort to make ATP. Those repeated rounds of glycolysis don't need oxygen. They're all good, but they do run out of something. And what they run out of is they run out of empty taxicabs or NAD+. Once all the NAD pluses are full of electrons and hydrogens, then glycolysis would shut down. You have to be able to make NADH as part of glycolysis. It's an important step where a phosphate is moved around. And so if you don't do it, the cell cannot do glycolysis. Well, what's the solution to that? In lactic acid fermentation, the electrons from NADH are put back onto pyruvate. And so they're dumped back onto pyruvate. They take the electrons from it, and they take the H plus as well. That's going to change pyruvate, because we've added stuff to it, into lactate. Lactate is an acid. It becomes lactic acid, and you feel it in your muscles after a while. There's some debate about that. That lactate or that lactic acid can then be sent to our liver to be processed later on. That's lactic acid fermentation. Some bacteria also do this as well, and they can give us sauerkraut and kimchi and other stuff. All right, yeast do a slightly different process where they're going to make alcohol. And we talked about this in class. One of the reasons they might make alcohol is it gives them a competitive advantage. It can kill other little microorganisms around them. So to continue glycolysis, right, to continue having enough NAD+, remember glycolysis was, we'd have to go way back, is here, where we're going to make pyruvate from glucose and make a bit of ATP. So for the yeast to do that, it's going to dump the electrons from NADH onto acetaldehyde. Acetaldehyde was made from the pyruvate, and then this acetaldehyde is going to be converted to ethanol, to alcohol. And so that's how yeast are able to regenerate the NAD plus needed to do fermentation. We didn't quite go over this very much at the end, but the carbon cycle is where this is all taking place, right, between plants and between different places that store carbon dioxide. So the atmosphere and in fossil fuels, the ocean, et cetera, these carbon atoms are moving around. You might have a carbon atom inside of you that was inside of a dinosaur. Right, and so we're impacting this by removing CO or removing carbon from fossil fuel storage and putting it into the air and increasing the amount of carbon in that reservoir. A flow would be the process of how it moves. And so the two flows that we studied here are cellular respiration and photosynthesis. All right, students, I wish you good luck. Keep studying hard. Let me know if you have any questions and crush it like a grape.